Hi, everybody. Here we are, Chapter 9, Biology, and we're moving on still with genetics. And so last chapter we talked about real specifically mitosis, meiosis, and then Punnett squares and, homozygous, heterozygous, all those kind of things, and just kind of how it all happens. This chapter in our books is titled Advanced Genetics. And so it goes into more of what we'll talk about as far as population genetics, but also mutations, things that can go wrong genetically, and some of the effects of those things. So first part of the chapter deals a lot with population genetics, genetic variation. And so the first thing to know are kind of some vocabulary words, basically, and gene pool is the first thing. And so a gene pool is all of the alleles, the genes that all of the members of the same species that are in a population together possess. And so, for instance, this would be like a herd of cattle. They are all the same species. They are within a population. They live in the same location. And so their gene pool is all of the genes that they contain. Now, they all have the same types of genes, all, for hair color and size and, all the things we've already talked about. But each member of that population has its own individual set of these same genes. And so that's what you're looking at as far as the gene pool actually goes. But remember, everybody's going to look different because of different combinations of genes, which means that the offspring of any of these animals will have the opportunity to look different too because of these different combinations. Okay, so speaking of, variations, these are genotypic variations. So remember, genetic differences between the individuals. They're in the same gene pool, and this is determined by those alleles. And there are three big things that cause this genetic variation. First of all, random gamete fusion. So remember that it is totally up to chance which sperm will fertilize the egg. And so each sperm that is being produced is slightly different from the other sperm because of crossing over and because of independent assortment. And so just totally random on what genes actually get put together. And like I said, that's based on that segregation and crossing over. And then the other one, which we'll talk about more as we go on through the notes here, are mutations. Every single living organism has mutations in its genetic makeup compared to its parents or even what that individual organism started at because of things that happen to the actual DNA. Like I said, we'll get to that. Okay, so the genetic information in a gene pool, so in our herd of cattle, if that's what we're talking about, is going to constantly change as individuals leave the gene pool. They, the cattle get sold off, or heaven forbid they die, or you get new cattle in. And so this change and these new genes coming in or out, this is called gene flow. And gene flow can help you figure out or even change the allele frequency, how often a specific gene shows up in a gene pool. So if an animal comes in, a cow comes into the herd who has a different coat color, then that will change the allele frequency. And then as that cow produces offspring and her offspring have that same new coat color, you change the allele frequency even more. And so it's just, basically kind of the comparison between what types of things you have. All right, so now these, still looking at the allele frequency and kind of how it changes, that is very often described in terms of genetic equilibrium. And if the allele frequency is fairly stable in a population, then you're at what is called genetic equilibrium. In very large populations, this is pretty much how it stays. So if you have a herd of cattle that has, 150 cows in it, then you're pretty much at genetic equilibrium. There may be some changes, but because you have such a huge number, it's not very noticeable. But if you have a herd of 5 cows and one of them is different, or one of them has a calf that is different, you can change that allele frequency. Now remember, equilibrium essentially means stay the same, but it's not actually equilibrium, because the population number could be changing. Individuals in the population could be coming in or leaving. You could also have mutations that are occurring in the genetic makeup, and so it's just kind of on the face of it, we're at genetic equilibrium. And then when you see genetic drift, this is when an allele becomes more frequent kind of suddenly due to random events. So this first one, the bottleneck effect. So this is when a large portion of the population dies. So for instance, you have your herd of cattle, and some kind of disease goes through the herd, and 50 of your 75 cows die off. Well, all of a sudden, the allele that gave the other cows the ability to fight off this disease and stay healthy, all of a sudden it goes way up, the frequency, because everyone else has died off. So that's a bottleneck effect. And then also sometimes you have a small group that breaks away from the main population and they start their new population. So again, with our herd of cattle, so say you separate them out and you want these 10 cows and a bull over in this other pasture, and you leave the others where they are, then if there is an allele in that smaller group and it is passed on as offspring are produced, then you increase a certain allele frequency. And so this is called the founder effect. These are hugely important in genetic research. It can help you determine why some animals are healthy versus not. It can help you just determine why organisms thrive in certain areas and some don't. And I know we always kind of think of this in terms of people or animals because we're really familiar with those, but they have studied this kind of data in plants and in bacteria, in all kinds of organisms, and they have lots of data for kind of how things change. Okay, so now so we talked about population in general. One of the things we talked about was that changes in the genetic code can happen through mutations. And mutations happen anytime the DNA is copied or transcribed or translated. It happens more often than you think, but your body's usually very good, or an organism is usually very good at catching it, particularly if it's something that is going to be super harmful, and that cell will be destroyed. But a lot of times the changes aren't that big a deal, and they don't really do anything harmful to the organism, so they just keep getting passed on. Some mutations just happen. They're just a goof up in the processes. Sometimes they're caused by a mutagen, and that's some kind of substance that causes a change in the genetic structure. The worst one, of course, that we think of is cancer. So things like skin cancer can be caused by too much sunlight, damage caused by that. So that's considered a mutagen. Okay, so now these mutations. So we're going to talk about two different types. First of all, you have a germ mutation. Germ mutations are in gametes, or in the cells that produce gametes. So that's egg or sperm. And so these types of mutations are passed down to the offspring, and every cell in the offspring is affected. And so this is something that you see, so like Down syndrome is kind of an example we've already talked about. So there has been something that has messed up in either the egg or the sperm, and when they combine, you end up with this extra chromosome, the extra 21 chromosome. Every cell in that child's body has an extra chromosome. It's not localized, it's everywhere. And so you see much more of an effect overall on a germ mutation. Somatic mutation, this is a mutation in other cells, the cells that don't make gametes. And this type of mutation generally only affects a few cells in the organism. So this is like, when you get, like a freckle that appears out of nowhere. That's a mutation, but it only affects those cells that get the darker pigments in it. Very, very rarely are somatic mutations as serious as a germ mutation, and they can be, but for the most part, germ mutations are you're stuck with it basically. Okay, so gene mutations, so the results of these gene mutations. So it's all about the nucleotides. Remember, the nucleotides are the little, the sugar and the phosphate backbone along with the base, adenine, thymine, cytosine, guanine, those guys. So it depends on them and what happens to them, and there are point mutations, which we'll talk about first. And this is when a single nucleotide, something happens to it. And there are 3 types of point mutations. The first is substitution. And this is when a single nucleotide is removed and replaced with a different nucleotide, or when 2 nucleotides like switch places. This causes problems because the new codon, the new set of 3 bases, very often or could code for a different amino acid. And so if you remember several chapters ago, we wrote out the big long list of letters, and we had like, A, U, G, A, C, G, A, U, whatever it was. And every third base coded for a different amino acid. And that chain of amino acids made a protein, and then that protein got used by your body. Well, if you switch one of those, then the protein, the amino acid gets changed, which means the protein very often is different. And so it can be somewhat of a problem, but also probably more frequently, your body adjusts to that and it makes the same protein anyway, just because of the way the DNA, or the mRNA in this case, is read. Okay, so substitution, not huge, but you still don't want it to happen. And then addition and deletion. In addition, sometimes this is called insertion. Just like it sounds, an extra nucleotide is placed into the segment. This causes what is called a frame shift. And a frame shift means that all the bases get moved down by 1 and the codons are changed. And so this is, so if you have that AUG, I don't remember what I wrote on the other page, but AAC, GA, GUA. Okay, so I've got my nice, sets of 3 here. But if I come along and I insert or I add in another letter, like right here, so the AUG stays the same, but now all of a sudden I have GAA, and then my next codon would be CGU. And so it completely changes the bases, or the codons, and so it can cause what is called missense, where a protein is made, but it's not the right protein, so it doesn't function. Or it can cause nonsense, which means just no protein is made, your body's too confused. And so that can be obviously pretty serious. Then you have deletion, and this is where you come along, and so now we have this. Let me erase this G out of here real quick. So you get the G, and so everything is kind of back to normal, or here's our 3 sets. But now I come along, and instead of adding something, I come along and I erase this A. So now my shift goes, so the AUG stays the same like it did before, but now I get a new codon of ACG, and I have this extra at the end. And so again, it can cause missense, get a protein, but it's not the right one, so it doesn't do anything, or it causes nonsense and no protein is created. So those very often are overcome in the system, but if they're not, it can cause some problems. So this picture kind of shows what I was trying to draw on that on those previous pages. And so if you're following along with your notes that you got from me, this is a different picture than what I gave you guys because I liked this one better. But anyway, so that first line is everything happens like it should. Totally normal. You get the right chain of amino acids, everything is good to go. Substitution, so notice in substitution, this little guy right here, oops, let's see. I think I'm erasing instead of trying to write here. So this little guy right here ends up changing things. So the DNA changes, you get a different letter there, which means then you have a different match to it on the mRNA, which means you get a different amino acid in your chain, which means potentially your protein is different. So could definitely cause a problem there. All right, so then the next, the insertion, which we called addition. So if this, this C gets added in, moves everything down by one, that's called the frame shift. And then notice you change two amino acids, which definitely has a problem, or definitely causes a problem for your amino acid chain, which causes change in the protein, which could cause big issues. Deletion does the same thing, and this time you actually, on our picture at least, when you add, or excuse me, you take out this little A right here, it's gone. Everything gets shifted down, and so you end up with 3 different amino acids, so a completely different protein. And again, your body's very OCD about the proteins it uses, and so that can cause big problems. Okay, so now those were gene mutations, and now we've got chromosome mutations. So now, what if we affect a whole chromosome? And so much bigger pieces of information potentially. All right, so first off, translocation. In translocation, you have the exchange of genetic material between 2 non-homologous chromosomes. Remember, when 2 homologous chromosomes trade information, that's crossing over, and we like that. That's why we end up with a mixed match of traits from your mom and dad. Like, then that's why you see things that your grandparents gave your, that kind of skip generations. So that is a good thing. But if the wrong chromosomes exchange information, then you have this translocation, and then they're not in the right place. The information's in the wrong place, and your body can't find it to make the right protein, which can cause some really serious issues. Deletion. In deletion, you just lose a complete segment of a chromosome. This also can happen during crossing over. If the chromosomes break apart and then they think they're going to attach to the other chromosome, and for whatever reason it doesn't happen, then that chromosome doesn't have the piece that it needs, and that piece that it does need is lost. Inversion is when a chromosome breaks off but then reattaches in a reverse order at the same position. This one is actually not that big a deal. Turns out your body can read the DNA backwards very often, and so as long as the promoter genes are there in the right place, then the RNA polymerase knows what to do, even if it has to go backwards. Okay, so now, so those were changes in the individual chromosomes. Now changes in the number of chromosomes is always caused by a problem called non-disjunction, which I should have highlighted that word for you, but there it is. And that's the failure of the chromosome pairs to separate. Either the 2 X's separate, like in meiosis 1, or the individual X separates 1/2 and 1/2 in meiosis 2. And so when this happens, you have 2 gametes that can be produced, and they have an extra chromosome, and then you can have, so that's one like this, we're down here where it has 3, or these 2 over here. We've got 3 chromosomes all of a sudden in there, or 3, a set plus 1, excuse me, and then you have 2 gametes that have 1 less chromosome than they should have. So they have, if we're talking about people, remember, you normally have, would have from your mom 23 chromosomes and from your dad 23. But so now say your dad gives you 24 and your mom is still giving you 23, then you have that odd number. Or if a mom gives you 22 and your dad gives you 23, so things don't match up. Okay, so when these altered genes, the N+1 or the N-1, when they combine with another gamete, so the normal, the offspring will be what is called aneuploid. Aneuploid means your number of chromosomes is just different than normal. So remember, normal we designate as 2N. That's called diploid. That's what you guys are. And abnormal can be that 2N plus 1, which is called trisomy, where you have all the chromosomes are diploid except one. That one chromosome, you have 3 of them, so that's where the trisomy comes from. The common one, which we've already mentioned a couple times, is Down syndrome. That is caused by having a third number 21 chromosome, and so its presence is what causes some of the different characteristics that you see in a person who has Down syndrome. All right, then another abnormal form, if you have this change in the number of chromosomes, is monosomy. So all the chromosomes are diploid except one, but this time you're lacking one. Turner syndrome, which is a syndrome that affects the sex chromosomes, is a female who just has one X. She didn't get the second X from either her mother or her father, and so still female. The side effects of this one, there may be some physical outward features. I believe Turner syndrome women have to be a little shorter, a little kind of stouter, not as much of like a feminine figure necessarily. They are usually unable to have children, but nothing super dangerous as far as overall health. Okay, so now other chromosome number changes. So now, what if you change the number of sets of chromosomes? And so this can also happen because the chromosomes don't separate, but now instead of just one not separating, you have whole sets not separating. And so if you have these unusual numbers of sets of chromosomes, you are called polyploid. And these only occur in plants and at least in organisms that can survive with this condition. Animals and people, you don't see triploids or the tetraploids. So a triploid is 3N, and so you've got 3 complete sets of chromosomes. All of these are plants, like I said, and there's a benefit to it. The plants themselves are usually larger and stronger. The fruit that they produce is usually bigger and better. So this is a sought after trait if you are a farmer of some of these types of plants. And so there are ways to do this genetically to change the different combinations. And it does help as far as the products go. All right, then tetraploids. So tetraploids are 4N, so 4 complete sets of chromosomes. Same characteristics as the triploids, basically. Produce is bigger, better. Plants are larger, and so some common tetraploid organisms are Irish potatoes, alfalfa, corn, certain types of wheat. And so again, it's a benefit in these types of plants as far as the outcome goes. Okay, so now mutation as it relates to cell growth. So if the mutation in the cell causes problems with the genes that control cell growth, one of the things that can happen is the cell just starts to divide uncontrollably. A normal cell has kind of like a personal space boundaries. As soon as they start to get kind of crowded, they stop growing. These abnormal cells just keep growing and start to push everything out of the way. They form an abnormal mass that is typically called a tumor. And these masses have 2 different categories. If it's benign, they are made of the tumors are made of cells that don't spread to other parts of the body. They stay localized. And a lot of times this is a little less dangerous depending on where the benign tumor is. but typically these, we kind of want this, you want to hear that something is benign because that means it's not spreading. Malignant, these are tumor cells that do spread to other parts of the body. We call this metastasizing. Generally, they will break away from the original tumor cell and travel through the bloodstream to other parts of the body, and then once they get there, they kind of take hold and start to again divide uncontrollably and spread there. And so this is typically what we call cancer. One just kind of a side note, leukemia is a form of cancer, but it doesn't involve tumors. It's just abnormal blood cells, and they cause problems. So if cell growth is affected, you have some bigger problems as far as how to deal with kind of shutting them down. Okay, so cancer itself is usually found to be caused by a mutation in one or more, I left out a word there, in one or more of the genes that work to prevent this uncontrolled cell growth. And so you basically have two genes that are working. There's really more than that, but these are the kind of the two big ones. Your proto-oncogenes, they code for a protein that tells your cells to grow. Normal growth, they can produce the growth factors. There's our hormones that tell the other cells to keep growing. So these are normal, the body needs to grow. But then you also have tumor suppressor genes, and these control or prevent uncontrolled cell growth. And they basically kind of checks and balance system. As the proto-oncogene is telling cells to grow, the tumor suppressor gene is kind of watching and making sure they stay within the right boundaries. And so the mutation is very often then called an oncogene. And this is basically just an overstimulation on cells. They just grow out of control, and it doesn't allow the cells to die. Most cells will reach a point where they just are worn out and they will just die. With this mutation, they don't reach that point. And this mutation is often caused by radiation, like sun exposure, different things. Tobacco smoke is a form. Different chemicals can do this. And so, they are the kind of overriding the natural genes. So when this cancer happens, the tumors form if the cell growth exceeds the strength of the ability of the tumor suppressor gene to shut it down. Okay, so almost finished up here. So genetic engineering. So what can we do as far as controlling the changes in the genetic code, or really simply figuring it out? And this is a huge field right now, but this is the study of genomics, and this is a study of genomes. And every organism has a genome, and it is the full set of genetic information. So for us, it's all of our DNA. What does our DNA say? And starting in the early 2000s, there was a big, huge push in the biology field and genetics to complete what was called the Human Genome Project, which basically was to map the sequence of DNA base pairs in a human to determine, like what the genes were for, what the phenotypes were from those genes, what the variations could be. And so in 2003, they finished this up. One of the main objectives was to gain information about diseases, like what starts it, where is the gene, is it what activates it, and so diabetes, Alzheimer's, cancer, and that's just a few of them. But this was kind of the start of people doing genetic testing, and then potentially like having surgery. Like for instance, Angelina Jolie was at this time kind of known for she was tested for the gene for breast cancer. They found that she did have it, and she had surgery and had these things to kind of preventative surgery in case this cancer did actually become active. And so anyway, so it's kind of a step forward in disease prevention. Okay, so now reading and kind of manipulating the DNA, how do you do that? And this is called genetic engineering. And so you are trying to manipulate the genes to do things other than their natural processes. And like I said before, anything genetics is just a huge field still. So just kind of be aware of that. But there are 4 things that we do that that are different than just like the mapping, just the genome project. So first off, reading the DNA. It would take forever to read all of the DNA in a cell. There's just too much of it. And so when you read DNA, you really are only reading very small segments. And you can do this more easily by using a restriction enzyme. And this is basically like a little pair of scissors, and it cuts out the DNA strand in very specific points to allow you to study, instead of thousands of base pairs at once, down to a couple of hundred. Sequencing DNA, this was a big part of the genome project. This involves determining the order that the nucleotides are in, and there are computer systems that you can input your data, input your samples, and it will sequence these pairs of DNA and then put that section where it belongs in the genome. It's again one of those kind of marvels of modern technology, but this is where we've gotten to, which is fantastic. Then we can make multiple copies of the DNA. We know how to use a polymerase chain reaction, which basically is a chain reaction, like so it just keeps going to make multiple copies of DNA segments. So once you've cut them up there with the restriction enzyme, and you just make a bunch of copies, so that as you experiment on one, then you have more to do different experiments on. And then last one, customizing DNA, and this is something we learned from viruses actually, but this involves putting in a new segment of DNA, and this is called recombinant DNA. And so you put this into the existing DNA to change the phenotype of the organism. So you're putting something in to a cell or into an organism in order to make it, in order to change its genetics just a little bit. Viruses do this with people all the time. This is what makes us sick. The virus puts its own DNA into our cells. We make what the virus DNA codes for, and it's the stuff that makes us sick. Luckily, your immune system takes care of it pretty quickly, and so you get over it. But that's basically the same process. Okay, so using this engineered DNA, one of the big ones and somewhat controversial uses is genetically modified organisms. And it's GMOs, and that's any organism that has been genetically engineered. Plants for years and years have been genetically modified to resist insects, to resist certain diseases, to produce more fruit, to kind of do whatever. Corn, sugar, soybeans, lots of things that we eat now could have been genetically modified. Lots of box labels will have non-GMO on it. Some if it's not labeled that, then you assume that it is in there. And there's some good things to it. We don't have as many chemicals on our plants because they are genetically able to defend themselves. And so that's one less expense, first of all, for the farmers, but also one less thing that we might ingest that could cause us to be ill. The cons are we don't really know the long-term effects of consuming these GMOs. They haven't been around that long. We have seen, though, definite indications that there could be some problem for certain people. The frequency of things like celiac disease, gluten intolerance and whatnot, became there's a huge spike in those cases. I believe in the mid-80s when we in the US first started using genetically modified wheat. And so those people that you may know, or you who have trouble eating gluten, it's thought that there is some kind of relationship between the genetic modification that we see in the wheat that pretty much we all are accustomed to versus the wheat before that wasn't genetically modified. And so there is a taste difference somewhat, but chemically how your body reacts to it is very individual. And that's why some people can eat all the bread they want without trouble, but some people, it can make them really sick. Okay, the next one, gene therapy. And this is basically changing a gene to help relieve a disease or a disorder. And you use that recombinant DNA that I was talking about before that is like part of the like a virus type thing. And so if you can put in a segment of DNA that will either help with certain symptoms or side effects of an illness, or even stop them, then you could really improve someone's quality of life. And so that's a pro. If you can block these symptoms and produce the right proteins, the healthy stuff your body needs, and you feel better, it's always good. The cons are either your body doesn't always accept the recombinant DNA, it just recognizes it's foreign and kicks it out. But also that recombinant DNA is very often not passed on, and so it's a treatment that requires lifetime treatment, so it's not just kind of a one-time deal. Okay, last but not least, DNA fingerprinting. This is where you can use little fragments of DNA, stuff from crime scenes, and match it to either a suspect directly, or you can use it to match to relatives, and so you can kind of see what happens. But they do this all the time now on CSI. They take a DNA sample and then they see if it matches a sample from whatever crime was committed. Sadly, they use it to identify the remains of people who maybe after an accident or something are just not recognizable. They can use it for people who have been murdered and have been dead for a long time. As long as there's still some DNA that can be used, then they can possibly trace it back. Also can use it to see some genetic disorders that are inherited. And so the big huge field of genetics, it's really super cool. We're learning more every day. And so as always, if you have questions, if this is confusing, talk to me about it, and I will see you in class. Bye, guys.